everyone, and welcome to Research Recap brought to you by Arbor Assays. Today's episode is a little different. Our guest's research spans not just one, but two studies on giant pandas, both of which use Arbor Assays kits extensively. This episode will focus on the first study, which established techniques and laid the groundwork for the second study that will be featured in the next episode. To kick things off, we'll dive into how the initial study came together and why those early choices mattered. Hello, everyone, and welcome to Research Recap brought to you by Arbor Assays. In this series, we dive into fascinating studies conducted by scientists around the globe, exploring how they're using Arbor Assays kits to unlock new discoveries. I'm Cassie Shoemaker, Sales and Marketing Manager at Arbor Assays, and I'm excited to highlight the amazing work being done in labs like yours, share insights, and take a closer look at the tools driving innovation and research. So let's get started. Today, I'm joined by Dr. Kirsten Wilson. Kirsten is from Edinburgh, Scotland, and studied human anatomy at the University of Glasgow as an undergraduate before undertaking a master's by research in reproductive biology at the University of Edinburgh. In her master's program, Kirsten had the opportunity to work on a project with the giant pandas at the RZSS Edinburgh Zoo and immediately knew she wanted to continue in that field of research. After her master's, Kirsten spent the next 8 years combining working in an assay facility full-time and undertaking part-time PhD in giant panda reproduction. She now continues to work in the facility after graduating in 2024. Welcome, Kirsten.
No, thank you for having me.
Yes, thank you so much for being here. What an exciting place to do your training and continue to work.
Yeah, it's a great place to live and work in. There's so much exciting research going on in Edinburgh. We're such a diverse research community here.
That's fantastic. Can you tell us a little bit more about what kinds of questions you hope to be able to ask and answer with this study?
So with my research, we were specifically interested in giant pandas. I mean, they're such a charismatic species. So, I mean, you just look at them and already it's exciting. But as soon as I started reading about their reproduction and seeing all the challenges that they face, I was really sold on conducting more research. So some of the different features that pandas show make it really challenging to monitor their reproduction. So one of the first things is that they're a monoestrous species. So they ovulate just once a year, meaning there's one chance a year to get pregnant. And that ovulation timing is very variable. So it happens in the spring, but there's a kind of 3 to 4 month window for that. And the timing is different between years and between pandas. So one panda is not going to be the same as the next panda. And after they've ovulated, so even if there's successful fertilization, the next challenge is that pandas undergo a delayed implantation or embryonic diapause. So once the egg has been fertilized, there's a pause on development. And this pause lasts typically between 3 and 5 months. And again, this is variable between pandas and between years. So there's a lot of unpredictability there. And at the minute, we don't know what the signal is in their biology that tells that pause to break and for development to continue. So that's kind of one of the ongoing things that we're still trying to work on. I'd hoped to solve it in my PhD, but yeah, that was too big of a question.
Yeah,
The final thing that's really challenging with pandas is they also undergo pseudopregnancy. So the behaviors that a female shows and also the progesterone profile. Now, progesterone in many species is known as a pregnancy marker. It increases during pregnancy. But in pandas, because of pseudopregnancy, that progesterone profile plus their behaviors are exactly the same in both non-pregnant and pregnant females. So what's typically a pregnancy hormone in some animals is not in pandas. So we can't use that to tell us if a panda is pregnant or not. So that's been a huge challenge, the three of them really, to try and unravel a bit more so that we can hopefully get some questions about their reproduction.
yeah, absolutely. It sounds like you had your hands full with plenty of questions to ask in such a beautiful but complex species. Can you tell us a little bit as you were getting going in these studies? You mentioned some of the hormones that have are there in pandas and not in others, and vice versa. So, can you walk us through the biomarkers that you chose to focus on and the ones that you were really diving into in this study, and why you chose some over others?
Yeah, so in pandas, we are primarily looking at their urine because we can't really take regular blood samples. So we're focusing on a non-invasive approach, which means we can really easily collect these samples. They're all collected from the floor of the pandas enclosures by the keepers. And actually, the urine's got so much information in it. So panda urinary hormones in pandas have been studied for over 40 years. So there is a vast amount of information out there. And before we started, estrogens and progesterones were the commonly monitored hormone metabolites in the panda urine. So I was really focused on the reproductive hormones, so really all of the sex steroid hormones. So for one of our papers, which we were looking for pregnancy biomarkers, I was actually still interested in the estrogens. So every study before us over the past sort of 40 years have said that estrogens remain baseline throughout pregnancy. There's absolutely no change. They don't bother monitoring them, and they had really actually only shown data from just a couple of females. And I was a bit like, are we sure that that's what they do?
Now let's confirm.
So I really wanted to confirm that. So I was very attached to estrogens. But one of the things we've been investigating was instead of using the typical creatinine hormone correction, so because urine's got different concentrations, that concentration means there's different hormone concentrations in each sample. So it completely can vary from day to day and even sample to sample within a day. So you have to correct for something. So most papers, in fact, all panda papers have looked at creatinine, but we instead were going to look at USPG, which is urinary specific gravity. And what we did find from that was seeing that the USPG shows a lot less variation than creatinine does in the samples. So when you then correct for that, there's less variation and it results in a bit of a smoother hormone profile. So we had tested this first with our progesterone, which we knew what profile to expect. And then so we could compare the creatinine and urinary specific gravity corrected data really nicely. So what I was looking at was, even though it's the same hormone, it's looking at it kind of in a different way.
Great.
Because estrogens are a pregnancy biomarker in a lot of different species. So I was so attached to that. I didn't want to let it go. I was holding on to that.
So, as you were looking at all of those different biomarkers that you described, you had a lot of different questions. Tell us a little bit about how the Arbor Assays kits kind of fit into that story, and how you were introduced to these kits in general, why you selected them, any insights you have about the brand.
So our lab, before I started, was already using the Arbor Assays kits. We'd been recommended them by other giant panda zoos across in the US. So we had been using them as well and had success. We had attempted to do some of our own progesterone assays, um, but we found they just weren't quite the same because there's so many progesterone metabolites out there that we were kind of even slight differences could give you a different profile because there's just a slightly different progesterone metabolite being picked up. So we wanted to stick with the Arbor assays. But once we started collaborating across Europe, because collaboration has been a huge thing in my projects, because pandas are loaned out in pairs. So each location only has one female and one male. So I was lucky enough to set up collaboration with Dr. Jelle Wouters, who was at the University of Ghent and now at Paris Dysat in Belgium. And she was really key in setting up a sort of, I guess, a European link of all of the pandas, because when I started, there was increasing numbers of pandas in Europe. And so using Arbor assays, because you ship worldwide, we can all use the same kits. And then if we're all using the same progesterone kit or estrone kit, then our data is comparable as long as we're following the same protocol. And that means that even though I did get scent samples from the pandas, I already had data there from the estrogens and the progesterone and PGFM. I didn't need to repeat any of those. So even if there's a tiny volume of sample available, I can make best use of that where it needs to be used. I don't have to waste time using it on something that's already been measured.
Yeah, that's wonderful that the kits allowed you that continuity across such a precious species and a number of samples that you had. I'm glad to hear that. Then you have your experimental setup. You have all of your plates run. Tell us a little bit about what you found.
We were looking at the urinary estrogens across the luteal phase and different reproductive outcomes. So with pandas, we split them into three reproductive outcomes. We've got pandas who are pregnant, pandas who we call pseudopregnant. So these ones were not mated or inseminated, so they are definitely not pregnant. And then we have non-births. So that is pandas who were mated or artificially inseminated, but didn't go on to give birth. So we can't confirm if they were pregnant or not. Um, and we looked at daily urinary samples, and we tested all of these on Arbor assay kits for estriol, and then we also looked at estrogens. So we looked at the E1G and the E1S because we've done some comparisons between the two, and actually you can use either. We also used the P4 and the PGFM. And yeah, we used that urinary specific gravity as our correction instead of creatinine. So I had really high hopes for estriol because it's a placental specific estrogen. So I really wanted estriol to work, but sadly there was absolutely no difference between our pregnant and our non-pregnant pandas. So I could write that out relatively early on. But luckily the estrogens, so the E1G and the E1S, showed a really nice pregnancy-specific increase. And what we do is we align all of our cycles based on the PGFM profile, which is quite specific in pandas. And it was after a PGFM spike that we saw a really nice increase in pregnant pandas and a very stable low level in the pseudopregnant pandas. So what we did from that was then we made a model that you could then compare your non-births and even ongoing cycles against, and found that we could use the estrogens as an indicator of potential pregnancy loss. So next, we then have the model we showed, and we validated that with a few more cycles that we've been able to gain, which luckily ended, there was one pregnancy and one pseudopregnancy, so it's really good to validate as well. And then we put on our non-birth cycles, and they showed really quite mixed profiles. Some of them are really clear and show a decrease, and we could tell quite early on that those were going to end up as non-births, or maybe the panda hadn't gotten pregnant. But some of them show a slight increase, but a premature decrease before we would expect if she had been pregnant. So they can be used for ongoing monitoring, which is really useful to be able to say in advance that a panda's not going to give birth, because it's quite a lot of resources go into monitoring pandas, not just from a lab side of things, but in the zoo itself, the keepers set up 24-hour watches, and it's an intense process
Yeah,
to
absolutely.
go through. So our main conclusions from this study were that estrogens do show a pregnancy-specific profile, but this is only when you correct for urinary specific gravity and not creatinine. The creatinine profile is the same between pregnant and non-pregnant females. So yeah, the papers were correct and that it is low, but they were correcting for creatinine and not USPG. And yeah, we can also use it in ongoing cycles. So yeah, it's really important to keep going along. And we also use it in combination with a number of other markers as well. So it's not just about one thing we're looking at. It kind of needs a combination, especially in the profiles that are maybe starting to look a little bit iffy. They're maybe looking like it might decrease, because you do get quite a lot of day-to-day variation in the urine samples. So we like to look at multiple things, and we found that estrogens really gave us a huge help in trying to determine if a panda was going to give birth or not.
Thanks for watching. Be sure to tune in for part 2 of this episode, where we'll explore the second panda study. We will also wrap up with some final thoughts on the implications of these studies and how Arbor Assays supports researchers around the globe.